Our next group is the euglenophytes, which is a subset of the euglenids, or euglenozoa. Basal members of this eukaryotic group do not have plastids. They are heterotrophic, typically living by preying on bacteria or other eukaryotes. The euglenophytes are the descendants of euglenids that acquired chloroplasts through secondary endosymbiosis. Compared to the cells we've been talking about, many euglenids are relatively large flagellates. Bear with me while I draw one out. Now, of course, to be green, a euglenophyte has plastids in it, and a nucleus, and all the things you might expect. I've drawn in two flagellae, one of which barely emerges from this pocket, and the other of which is long, and the cell waves it around to swim. There is something else in there with the axoneme, which is in yellow, coming out of the basal body there. If you take a cross section, this axoneme is joined by some sort of rod, whether it stiffens it or provides contractile properties. I don't think I remember, if anyone knows. At any rate, that's a characteristic innovation of the euglenozoa, and they indeed use their leading flagellum in many interesting ways, both for flagellar swimming and also for gliding motility. In particular, the lab students will see that in one of the non-photosynthetic representatives that is easy to get in culture, Paranema. Paranema is a gliding predator. It's happy to feed on other eukaryotes, in particular euglena, as it happens. One of the other characteristics is also really well developed in Paranema. The corrugations here on the surface represent the presence of not an external scale coat or a cell wall or something, but a system of interlocking strips. It's just called the pellicle, but the simple name belies a remarkable function. So obviously this goes all the way around. I'm not going to draw in every strip. These strips are arranged often longitudinally along the cell, and they are made of some sort of protein, and they fit together kind of like this, with a hook and notch kind of mechanism. there are microtubules associated with the pellicle strips. I don't think the exact mechanism is known, but if, imagine these strips extending back that way, they slide back and forth relative to each other. And as they do this, they deform and twist the cell shape in quite a dramatic way. Parenchyma in particular writhes and squirms quite delightfully, especially as it tries to chew its way into some hapless cell. So again, this is referred to as the pellicle. These pellicle strips are a unique euglenid sort of thing. These plastids have, wrong color, have 3 membranes around them. Again, I think this doesn't really mean what people once thought it meant, but nevertheless, that's the way they are. Students in the lab will also see Euglena gracilis, which has a number of important roles in the history of biology. Some Euglena species, by the way, are quite large, up to 200 microns long or so. This one is not so big. It is an autotroph. It does not squirm around quite so much as Parenchyma does. This name, Euglena, comes from this eye spot, a red carotenoid-rich patch that is juxtaposed, again, to a photosensor alongside the flagellum. So there's a light sensor in here, a filter here, and as the cell moves and rotates, it can estimate the direction of a light source and respond with a change in the flagellar beat or perhaps other cell traits accordingly. I read somewhere that Euglena was named by a 19th century zoologist, I forget who, inspired by a line of Ralph Waldo Emerson's. So John Locke, the philosopher, had famously described the state of human knowledge in rather depressing terms. He imagined himself stuck in a box with just a slit through which the light of illumination came through. And Ralph Waldo Emerson said in typically poetic fashion that instead he was a naked eyeball floating in space, open to the illuminating experience. And whoever discovered Euglena thought, aha, I've got one of those. So its name means good eyeball. Cell division in Euglena takes place by furrowing from this flagellar pocket. Furrowing follows duplication of the flagellae, but also duplication of these pellicle strips. Through some choreography, they duplicate themselves starting at one end and go all the way down. This strip doubling precedes mitosis. The basal bodies, after flagellar duplication, move to opposite sides of the very large nucleus, which has this permanent nucleolus in it. So the basal body is still associated with the flagellae, sort of string the nucleus between them. The nucleus does not break down. Chromosomes condense in there. and seemingly arrange each their own independent bundle of microtubules, a bipolar bundle. Those all somehow line up within there. I don't think anyone knows how. And then this nucleus as a whole divides during telophase as furling comes down to divide the whole cell. Why am I telling you this? Does it really matter that you know how some weird group of protists divides? No, of course not. The point is not to instill in you specific knowledge of the division mechanisms for each group of eukaryotes, but rather to emphasize that these things that we talk, these processes that we talk about, cytokinesis and mitosis, we know them well in some cases, but we also know very well for some cases that they differ greatly from the stereotypes that we know. So each eukaryotic group may have its own mechanism for mitosis, its own mechanisms for cytokinesis, which in some ways look utterly different, at least superficially different, from the mechanisms that we know of in animal and, say, land plant cells. Now we get to the cryptomonads, one of my favorite flagellate groups. And most of these are cryptophytes, but not all. This group includes some basally branching members that don't have plastids and that still live by eating bacteria. Also a sister group, the catablepharids, that are heterotrophs. So the key thing about this group, they're very definitely secondary endosymbionts. And their endosymbiosis very clearly involved a red alga for two reasons that I'll outline after I draw one. They're typically fairly small flagellates. With two slightly different flagellae decorated with hairs. They very often have a contractile vacuole to expel water, especially forms that live in fresh water. The vacuole fills and empties to get rid of excess fresh water, excess water that comes into the cell and might threaten to burst it. They have some sort of proteinaceous covering to the cell, not a cell wall. It's referred to as the periplast, which supports and stiffens the membrane, but doesn't provide any real barrier to, say, digestion by a predator. Of course, there's a plastid in there, or two sometimes, but they're not green. Well, okay, some of them are green or blue-green or something like that. These plastids retain the characteristic accessory pigments of the red algae. that help them to harvest light, even in dim light. More than that, these plastids have a so-called nucleomorph, something that looks like a nucleus inside the plastid. And in fact, this is a eukaryotic nucleus, or was once. It contains the remnants of what's clearly a red algal genome. So these cells, of course, there's a mitochondrion in here somewhere, and the mitochondrion has its own genome. These cells harbor a nuclear genome, the genome of the mitochondrion, the genome of the plastid, and the genome of the red algal cell that they originally engulfed to establish a secondary endosymbiosis. That's a lot of negotiating, isn't it? Okay. Cryptomonads have one other characteristic feature that I'm particularly fond of, lined up behind the flagellae along this gullet. And I should mention the gullet in heterotrophic members of the group and in cataplepharids is where they engulf prey items. Lined up along there are these little boxes. These are ejectosomes. Within each of those little boxes, which is in fact actually a secretory granule, a product of the Golgi apparatus, is a coiled ribbon. When one of those vesicles is secreted, that is, when it fuses with the plasma membrane and joins the outside world, this coiled ribbon inside there uncoils rapidly and rolls itself into a tube. So this coiled ribbon transforms rapidly into a long stick. Kind of like the so-called Chinese yo-yo, except that it isn't reversible. These ejectosomes are the cryptomonad's principal way of getting out of someplace in a hurry. If it's irritated or mishandled or squished a little bit, it will fire off some ejectosomes. And if these sticks hit something, then this cell takes a rapid jump of about the length of the ejectosome, which is typically 30 microns or so. They have a certain number of these cannonballs in them, and that is, again, probably their principal defense against predation, since the paraplast doesn't protect them from much. Back to that in a minute. If you look closely at the relationship between the nucleus, which remember is surrounded by its own double membrane with pores and so on, and the plastid, they appear to be quite intimately joined by the endoplasmic reticulum, which is continuous, as it is in every eukaryote, with the nuclear membrane. So in a way, you could think of the nuclear membrane simply as a special compartment of the endoplasmic reticulum, or you could think of the endoplasmic reticulum as an extension of the nuclear membrane. Whatever. It doesn't really mean anything to say it either way. They have distinct functional roles. Nevertheless, this nucleomorph is tucked in there like that, and then the chloroplast itself let's not forget those thylakoids. It's this here that is the actual pigmented photosynthetic bit. And again, it's red because these cells retain the accessory pigments, the protein-based fluorophores that augment the light harvesting capacity of chlorophyll. What's remarkable here is the number of membranes that have to be crossed in order for gene products encoded in the host cell nucleus, or vice versa, to get into the plastid. And there is another group of eukaryotes not related to the cryptomonads, another group called the chlorarachniophytes, also secondary endosymbionts, except they got their plastids from green algae. And the reason I'm pointing these out here is that they also retain, in that case, the green algal genome as a nucleomorph. And we will read a paper which addresses, well, what's inside the nucleomorph? Cryptophytes are, they're very widespread, first of all. I forgot to mention, they're found in essentially all the waters of the earth, even some of the frozen ones like ice or in brines. The Salton Sea, for example, this hypersaline artificial body of water in Southern California is home to a lot of cryptophytes that bloom there. They've probably been around for at least 1 billion years, and there's some reason to think that the cryptophytes, the cryptomonads, are the first group of eukaryotic heterotrophs that figured out how to conduct this secondary endosymbiosis, and that many other supposedly secondary endosymbioses are perhaps even tertiary endosymbioses. They may have all gotten their plastids from the cryptophytes. That's a hypothesis, not something that's known. At any rate, these are naked, digestible cells. The way most animal biologists meet cryptophytes, if they meet them at all, is as a laboratory food for animal larvae. Those of you who are taking the lab have been feeding Rhodomonas, a cryptophyte, to animal larvae because they are particularly good digestible food. They are cryptic largely because they burst easily when people fix them to do counts of what's in the water, and so they are lost from samples easily. Because they retain the accessory pigments of the red algal plastid donor, let's call it, there's a good case that they can probably continue photosynthesis effectively in much lower light levels than many other algal groups can. That is, they can photosynthesize at greater depth in lakes or in the ocean at depths where nutrients are not limiting. Throughout most of the ocean, the surface waters are nutrient poor because other algae have taken all the nutrients down, have drawn down all the nutrients that are available there. Cryptophytes may be able to continue to proliferate in much deeper water and therefore have access to nutrients, in particular phosphate, that would otherwise limit their growth. These unarmored, almost vacuole-free and phosphate-rich cells may be an especially good food for animal larvae, as it happens. Our next group is the haptophytes. These cells also got their plastids from red algae or possibly from a cryptophyte. They're usually roundish with two smooth and equal flagella. Plus a third appendage called the haptonema. So these two are flagella. This thing is their own special sort of sticky outie. The haptonema is a membrane-bound microtubule-based structure like the flagellum, but in cross-section, if this is the membrane, there are, excuse me, I should color code those. There are 7 microtubules in there, singlets. Surrounded by a sheath of endoplasmic reticulum. There are some other proteins in there, notably this assembly contains centrin, a calcium binding protein that I've mentioned before, and centrin in response to calcium changes shape dramatically. Centrin can form polymers and link things together in various ways. The endoplasmic reticulum is the cell's store of calcium, and the haptonema might behave abruptly by doing things like bending, perhaps not unlike a flagellum, or even coiling up. The haptonema is involved in cell behaviors in haptophytes that actually have one that include attachment, capture of prey. Some cells appear to be able to grab things with the haptonema and then deliver it back to the cell body by contracting or coiling it up. Inside here, of course, there's a nucleus. For reasons I can't explain, it is often a rather rectangular nucleus, often a great deal of endomembrane. This is supposed to be stacks of the Golgi apparatus. And characteristically, golden plastids. These cells generally have scales produced within the secretory pathway and then exported to cover the cell surface. The most well-known form of these scales is as carbonate mineralized ones called coccoliths. The haptophytes include the coccolithophores, that is, cells that bear coccoliths. In the scanning EM, these are the scanning electron microscope. These are the cells that look like they've clothed themselves in paper picnic plates. or something like that, although some coccoliths can be even more elaborate. The coccolithophores, some of them, are so prolific, so abundant, form such great blooms that they are the cells you can see from space. Particular coccolithophore Emiliana Huxleyi is the most is the cell most commonly responsible for that, for such blooms. Looking again at the nucleus to chloroplast connection, the nuclear membrane is again contiguous with so-called periplastidial endoplasmic reticulum. So if we have the nuclear membrane there, this double-layered, pore-encrusted compartment, the endoplasmic reticulum, again, is an extension of that, or at least contiguous with it. Vesicle traffic from the endoplasmic reticulum to the Golgi apparatus constitutes the secretory pathway, as we'll talk about later. One of the things I want to highlight here is what's going on in this rather crudely drawn stack is that, oh, where'd the brown chalk go? Whatever scales they make, proteinaceous or calcified, are assembled within the secretory pathway. They are secretory products. If they're making great big wheels, for example, as many coccolithophores do, they have to be put together in parts and calcified internally. And then exported to the outside world intact. This manufacturing process in the secretory pathway is one half of what I want to illustrate. The endoplasmic reticulum, similar to the circumstance in the cryptophytes, although slightly different here. The nuclear membrane is contiguous with a perennial favorite among vocabulary lists, the PER for periplastidial, meaning it surrounds the plastid, endoplasmic, reticulum. Again, it's within this double sheet of the endomembrane system that the actual photosynthetic organelle, the plastid, is housed. So again, there are 4 layers here. So what? These 4 layers must be transited by anything going from the cytoplasm, where all protein translation takes place, to the chloroplast. I cannot show you, this is addressed to students taking the lab course, I cannot unfortunately show you a haptophyte that has a haptonema. If I could, I would. The only haptophyte that I can show you is a really utterly simple and boring one, isochrysis, which is commonly grown as a food for oyster larvae and things like that. These are pretty small, round, boring-looking cells. They don't, in fact, have a haptonema, at least not one that I can see. They are a part of the same group that includes coccolithophores, but they themselves do not have mineralized scales. In many species of haptophyte, vegetative growth in a haploid and motile state, so that is flagellated motile cells, alternates with a diploid state. These might fuse to make a zygote. This diploid might even grow larger. It is, it is this stage that blooms as coccolith-bearing non-motile planktonic algae. And of course, meiosis connects these but these can both proliferate vegetatively as well. What are these coccoliths all about? Are they armor? Do they help the cells sink? Who knows? One suggestion has to do with the chemistry of photosynthesis. As I wrote down earlier, photosynthesis means fixing CO2 using the enzyme Rubisco with the addition of that happens, two water molecules and light to carbohydrate water and oxygen as a waste product. CO2 is scarce nowadays, so it'd be nice to be able to use carbonate instead. Bicarbonate utilization means you gotta so this is 2 bicarbonate plus protons. And we can get out of that two CO2 and 2 waters. Now the calcification reaction looks like this. I should say I'm copying a scheme which I took out of the uh, the Graham algae textbook. If you want to follow this explanation in words, where this is headed is we're going to look at all these chemical equations together and cancel things. So calcification means taking a CO2 and a water molecule and a calcium and reacting them to get out calcium carbonate and two protons. At least that's one way of looking at it. So now we're just going to go through and cancel, right? Those are going to cancel out. We can cancel one of those CO2s and the other one and those waters, right? And there's another water that we can cancel. And what you're left with is a net reaction that looks like 2 bicarbonate plus calcium forming calcium carbonate. Excuse me. Carbohydrate. And oxygen. The point being to look at this in the context of the cell, which needs to conduct photosynthesis inside the plastid, needs to concentrate a carbon source, inorganic carbon in this case, and that means getting CO2 in the carbon concentrating mechanisms can achieve a high concentration of carbonate because it's trapped by membranes, unlike CO2, which is not. And the point here is that the coupling of calcification inside the secretory pathway as these cells make coccoliths may be part of this mechanism for concentrating carbon dioxide, may be part of this mechanism for concentrating carbon dioxide within cells for the sake of photosynthesis. It happens that these calcified scales might make the cells heavy. They might provide a source of carbonate that can be mobilized for the sake of photosynthesis. They also might provide something like an antacid tablet that deters or limits predation, because of course, whenever some unicellular thing swallows a coccolithophore covered in a bunch of chalk, basically, that chalk is going to buffer the digestive processes in the lysosome inside the little bags of death water that most cells use to dissolve phagocytosed food particles. So perhaps coccolithophores are also covering themselves in antacid tablets that jam the digestion of their predators. Wouldn't that be tricky? The last group we're going to consider is the dinoflagellates, which surely are close to the top of almost anyone's list of the strangest protist group. Dinoflagellates come in all sizes in a variety of shapes, but the standard thing is something like this. An epicone divided from a hypocone by a cingulum, or a girdle, if you will. The cingulum contains a singular or transverse flagellum organized like a screw, organized helically and coming around this side. Dinoflagellates typically have two flagella. The other flagellum, also emerging from this slot, is the trailing or longitudinal flagellum. The cells swim that way, so in this case the flagellum is trailing, as in our cells. This slot here is, by the way, called the sulcus. Many dinoflagellates are armored in a characteristic way with cellulose plates. The plate formula may be diagnostic for a particular species. I'm just drawing some random set of polyhedrons, or polygons, excuse me, obviously, not any particular configuration that I know of. Kind of made a mess of that, but we'll stick with it for now. The two flagella work differently and are quite innovative compared to other flagella that you've seen. For one thing, this longitudinal flagellum, although it often beats sinusoidally, can coil itself up rapidly, retracting into this slot, into the sulcus, perhaps all the way up. And it does so because, along with the axonemal microtubules, there is a fiber of centrin that rapidly can shrivel that long thing up into and stuff it in a pocket. So imagine that within the membrane of this flagellum is this contractile rod that, when it tightens up, bunches it up like a window shade. All of that would be wrapped inside membrane. The transverse flagellum also has this propensity to change shape, also due to centrin in there, presumably. The transverse flagellum, not only is its beat rate variable, but because it can bunch up or relax and spread out within there, it's the other characteristics of its beat also vary. The pitch of the helix varies in particular, and it can tuck it in or out of this slot. The propulsion of diatoms is in part due to the longitudinal flagellum, but also by coiling this thing up in a helix and sticking in a slot, they have achieved what many animals do, or many ciliates do, by using many cilia together. They have a little thrust maker sticking out of that slot at every position along the girdle band. The armor plates, if you look at a section of the cell, are inside, if they exist. Not all dinoflagellates are armored. If this is the plasma membrane out here, the armor plates are inside large membrane-bound vesicles called amphiesma or alveoli. So this brown object is supposed to be that cellulosic armor plate. This theca, the combination of the vesicles and the armor plate within, can be shed rapidly by fusing all these membranes together. In response to various challenges, many dinoflagellates will rapidly insist shedding the theca in just this way. In fact, some very inconvenient ones that one of my brothers used to work on have the unpleasant habit of shedding them when they're exposed to bright blue light, the kind that you would use when you're using, say, a confocal microscope to see what's inside one of them. Anyway, dinoflagellates come in a variety of shapes. Besides this, some form chains in which these little sumo wrestler type things are joined one to another. The chains are usually fairly short, unlike, say, chains of diatoms. One in particular that I keep as pets takes the hypocone and epicone to an extreme and makes itself into a swimming thorn. This is serratium, which comes in quite a variety. Others twist that general body form up in a variety of ways. Those don't depart really from this general plan, but some do. In the lab, students will see pyrocystis, which makes giant balloons with an island of cytoplasm in the center connecting to a very distant cell surface by tendrils. Pyrocystis is one of the highly luminescent dinoflagellates. There are many luminescent dinoflagellates. Around here, the optimistically named Noctiluca is not among them, although it is elsewhere in the world. Noctiluca is also a big balloon with an island of cytoplasm at the center of a big vacuolar space. Noctiluca, unlike Pyrocystis, is not content to just sit there. It has elaborated one of its flagella. The other one is flopping around in there, having lost the cingulum. It has elaborated one of its flagella into a tentacle with which it captures prey and stuffs it inside and into food vacuoles. So they have quite a variety of cell shapes. This is certainly not exhaustive, but those represent things that I think I have a reasonable chance of showing you in the lab if you are taking the lab course. Dinoflagellates multiply as haploids, by and large. Environmental conditions prompt differentiation into gametes, which may or may not be distinct, except that they would fuse to make a zygote. In the cultures of serratium, one may find that if the vegetative cells are this size and filled with golden plastids, as far as I can tell, some fraction of those are effectively female gametes, and then there are some small colorless cells swimming around amongst them. Those are the male gametes. I have watched and watched and watched until I've fallen asleep with my eyeballs resting on the eyepieces, and I have not yet seen them actually fuse. I would like to see it actually happen someday. Dinoflagellates, of course, have their own special form of mitosis called dinomitosis, in which the nuclear envelope never breaks down. Chromosomes condense within it toward one side, but, and here's the weird thing, the chromosomes are permanently condensed. They condense a little more during mitosis. Another fun fact about dinoflagellates, is that they do not use histones to condense their chromatin. Excuse me, I need to get these a little closer to the nuclear envelope because what they apparently have is an envelope spanning kinetochore by which they attach to what? Well, to some sort of spindle, of course. The poles of the spindle over here are connected by bundles of microtubules. These never enter the nucleus. Instead, the kinetochores hook up to these microtubules and are somehow eventually sorted along the elongating spindle, which frequently ends up boring a sort of a tunnel or several tunnels through the nucleus. At anaphase, imagine all of these chromatids being separated from their sisters along this sort of rail system. Why were my brother and I shining intense blue light on these? Why? We wanted to see how this works for ourselves because we don't get it and we still don't, because they aren't terribly cooperative with light microscopy the way I know how to do it. At any rate, so dinomitosis is very different from other eukaryotes. Although many dinoflagellates are photoautotrophic, many others are predators that hunt and subdue their prey. There are also many parasitic dinoflagellates. Of course, parasitism is often predation with a very different size ratio. The dinoflagellate feeding strategies include engulfment. So many species of Serratium, for example, are reported to sidle up to and swallow a prey cell. How they open up their armor to do that, I don't know, but that's the idea. Again, I have not witnessed this in person. I already mentioned Noctiluca uses its highly modified flagellum to capture prey. Noctiluca can get to be as much as 500 microns in diameter and can take even animal larvae if they aren't careful or swim fast enough. So when you catch Noctiluca in plankton samples, they're sometimes quite abundant, and you will find, in addition to diatoms, you'll find small animal larvae, ciliates, and various other prey items inside food vacuoles. These cells can even distend out greatly. I should mention that in Noctiluca, when they undergo sexuality, they differentiate into gametes that have this dinoflagellate type form. They make small, a big cluster of very small gametes down on one end of this balloon eventually. Some dinoflagellates engage in a kind of suctorial feeding in which a cytoplasmic protrusion emerges from the sulcus and targets some hapless prey item as they suck up their victim. Most remarkable of all, so that's the suctorial mode. This is engulfment. And then there's what's known as paleo feeding. Pallium is a cloak. This is perhaps the most remarkable of all. Many of the heterotrophic dinoflagellates that we catch in the plankton are out there looking for a very large prey item indeed, such as a nice big spiky chain-forming diatom like Chaetoceros. How are they going to deal with that? Spikes everywhere. The total volume for this colony that's greater than the putative predator. What this dinoflagellate does is truly remarkable. It begins somewhat like this suctorial feeding process with a bundle of membrane-bound protoplasm squirting out of the sulcus, but then it extends as a transparent veil, often over this entire thing, wrapping up every spine in this pallium. I have watched this happen. They're very touchy about it, but this dinoflagellate will swim around with this chain of diatoms enclosed within this veil of membrane and cytoplasm, and these die one by one, and the dinoflagellate sucks up their contents. Even many plastid-bearing dinoflagellates depend to some extent on prey capture. Even if they have the means to photosynthesize, it may not provide all of their nutritional needs for growth. And this may be why, in addition to their variety of feeding modes, dinoflagellates provide the clearest illustrations of tertiary endosymbiosis, including cases in which they house plastids of multiple different origins, or where there's clear evidence that they have replaced the plastid they once had with plastids from another lineage entirely. You know, I used to think dinoflagellates, the dino is got to be like the dino and dinosaur, right? Terrible lizards, terrible flagellates. Sadly, it turns out the dino in this case just means spinning. As they swim, dinoflagellates characteristically rifle through the water because of the action of the transverse flagellum.